Now today we are organizing the international webinar series from the Department of Microbiology of Hooghly Women's College on the topic an approach towards good health and well-being. Now, firstly, I would like to request Dr. Shamulina Goswami, IQAC coordinator, to say a few words. Now it's over to you, ma'am.
Thank you. Thank you, Piyali. And good morning, everybody. I convey my deep gratitude to all on behalf of my college to the first day of 2 days international webinar, an approach towards good health and well-being, organized by the Department of Microbiology in collaboration with Internal Quality Assurance Cell of Hooghly Women's College. In such an august virtual gathering, we are glad to have with us most eminent and esteemed personalities from different institutions of India and abroad. It's my pleasure to extend a cheerful welcome to you all. Health for all people all over the world is an important part of sustainable development. The World Health Organization defines health as a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. This is consistent with the biopsychosocial model of health, which considers physiological, psychological, and social factors in health and illness and interactions between these factors. It differs from the traditional medical model. The WHO definition links health explicitly with well-being and conceptualizes health as a human right, requiring physical and social resources to achieve and maintain. Well-being refers to a positive rather than neutral state, framing health as a positive aspiration. This definition was adopted by the 1986 Ottawa Charter, which describes health as a resource for everyday life, not the object of living. From this perspective, health is a means to living well, which highlights the link between health and participation in society. A holistic approach to better health will require ensuring universal access to health care and to making medicine and vaccines affordable. India's health system still facing multiple challenges. There are significant inter and intra-state disparities in outcome, and socio-economically backward groups are especially vulnerable to gaps in healthcare access. However, well-being as a concept is much broader than this and has been debated since the 3rd century BC. The philosopher Aristotle came up with the concept of eudaimonia, the contented state of feeling healthy, happy, and prosperous. Health and well-being are essential elements to increase and or maintain employee performance, productivity, job satisfaction, and engagement within the work environment. The current pandemic situation causing greater disruption is likely to have major impacts on mental health. We can see how rates of anxiety, depression, post-traumatic stress increased in the wake of COVID outbreak. I would like to convey my greetings to Dr. Abhishek Boni and Dr. Debushmita Mukhopadhyay, the distinguished speakers of today's webinar, for taking time out and being here. Once again, I welcome all the faculty, scholars, and students for their participation in such contemporary and thought-provoking issues, and request your active participation. Thank you. I would like to pass on to over to our principal, Dr. Shima Banerjee.
Thank you, Shamulina. Good noon, friends. Today we are all assembled here to participate in an international webinar organized by the Department of Microbiology in association with IQAC of the college. In this current scenario of COVID-19, we are not only socially apart, but also we all are suffering from mental distress. This COVID-19 has affected us in every aspect of life. The dynamic and energetic young generation is suffering the most. They are unable to attend their educational institutes and also sports for nearly last 6 months. We still have no idea about how long this situation will persist. In this context, Hooghly Women's College is conducting a series of lectures and webinars to motivate the students and faculty members. This will also inculcate and nurture their knowledge. Today, the Microbiology Department of Hooghly Women's College has organized a webinar on the topic An Approach to Good Health and Well-Being, which is very much relevant in developing the inquisitive thoughts of the young people. Only good health can resist any type of physical hazard. Hope all the participants, along with the faculty members, will highly enjoy the webinar. I wish a grand success of the webinar. Thank you all once again, and over to Professor Doel Roy Nag. Please, Doel, proceed.
Good afternoon, everybody, and thank you, Shamiluna Di and Principal Ma'am, for your wonderful introductory speech. So today I would like to introduce the first speaker of the today's webinar, that is Dr. Abhishek Banik. Dr. Abhishek Banik has done his BSc from Vivekananda Mahavidyalaya, Bardhaman. He has completed his master's from the Department of Microbiology, Bardhaman University. He progressed with his research work from ICAR, National Rice Research Institute, Odisha. He has also worked as a postdoctoral scholar at the Department of Plant Sciences in the University of California. He has his research experience on the topics which has a great impact on the field of agriculture and ecology. The topics are host microbe interaction, microbial diversity, bioremediation, biological nitrogen fixation, bioimaging, and cytostabilization. He has been awarded with various prestigious awards in a very short span of life, of his still now short span of time. He has been awarded with the Best Young Researcher National by IRDP Awards in the year 2018. AMI Young Scientist in Agricultural Microbiology by the Association of Microbiologists of India in the year 2019. He has also been awarded with UGC Startup Research Grant on this year, that is January 2020. He has 25 publications till now, and the time is too short today to discuss them in details. So today, Dr. Abhishek Banik will deliver his speech on the topic, Exploration of Plant-Associated Indigenous Microbiota for the Sustainable Agriculture. Thank you. Over to you, sir, Abhishek Banik.
Thank you, ma'am, for the generous introduction. Should I share my slides?
Yes.
Just a minute. Yeah, just a minute. Is it visible? Should I begin? Okay. Thank you, madam, for the generous introduction. Today, I'm going to discuss about exploration of indigenous plant-associated indigenous microbiota for sustainable agriculture. So, if you see, there are certain layers for plant microbiology. As I am working as a agriculture microbiologist, and my research experience and all the expertise on agriculture microbiology. So today I'm going to discuss about three topics of agricultural microbiology. First one will be beneficial role of endophytic and epiphytic microorganisms of rice ecologies. Second one will be microbes mediated phytostabilization, that will be in groundnut ecologies. And the third will be development of a Bacillus thuringiensis based biopesticide, that will be on tea ecologies. So currently, if you consider nowadays, Indian agriculture is facing several problems. Of them, I have taken three. I am basically doing research on three of them. First one is the nitrogen contamination. So 60% of the nitrogen that we are using in fields, that generally lost due to ammonia volatilization, nitrate leaching, denitrification, and rainwash. Subsequently, this ammonia, this nitrogen, when is got contaminated in groundwater, it creates several diseases in human beings. Subsequently, another thing is that when this ammonia, one have to use this ammonia again and again in field condition, so this ammonia get lost, and each and every year farmers has to use this urea to supplement the plants. So every year it adds the recurring cost. Additionally, when farmers use ammonia or the nitrogenous fertilizer in certain higher amount, it totally destroys the soil microbial dynamics. Additionally, this nitrogen fertilizer such as urea, it works as a magic bullet. Like if they are using excess amount of nitrogen fertilizer, within couple of days the plant will be totally greener, and the more greener means it will be attracted by different kinds of pests. Additionally, I'll also discuss some topics about the metal induced changes in the plants, because most of the agricultural field currently under invested with different kinds of heavy and transition metal because of the industrialization and different anthropogenic activities. So when these metals get taken up by the root system of the plant, they alters different physiological activities, and they ultimately it results chromosomal aberration. It reduces the germination of the seed. Additionally and subsequently, the result would be reduced crop production. So in the first topic, I will discuss about nitrogen fixing epiphytic and endophytic bacteria, and how we are doing this experiment and what is their benefits. So if you see the lifespan of a rice plant, it has three stages. First one is the vegetative stage, second one is the reproductive stage, and third one is the ripening stage. So the rice plant invests around 20% of its photosynthesized carbon at pre-flowering stage, at this particular phase. So they exudate that photosynthesized carbon in the form of amino acid, different kinds of sugar, alkaloid, flavonoid, and these compounds generally exudates from the root. As you know, the soil is very nutritionally poor, so soil microbiota, they used to fight for each other for a very minute amount of nutrients. So getting that huge amount of exudates that is coming out from the root, different kinds of soil microbiota get attracted towards the root, and some of them first they just come to the rhizosphere region and colonize on the root surface, and some privilege among them can enter into the root system, and from the root they can go to the stem and the leaf. So these are basically the endophytes. apart from endophytes, I have also done little bit work on phyllosphere and phylloplane organism. Phyllosphere means the close vicinity area that is closely associated with a leaf, and phylloplane means the microorganisms that is present on the leaf. So what are the advantage of these endophytes? Endophytes are basically, they are normal the soil bacteria. They are highly motile. When these root exudates act as a chemical signal, they just track those root exudates, as it can serve as a very good sole source of carbon to them, and some of them are privileged to enter inside the system. And the benefit of entering into the system is, in rhizosphere the microbial competition is far more. So when one bacterium or one group of bacteria is able to enter inside the root, their competition is very less. Secondly, the advantage of using endophytic organism is that they can compete with the pathogen. How? Simply, if endophytes, first they can colonize on specific pockets, specific receptor of the plant where those pathogen target. So the population matters. If the beneficial organisms first enhances their number, they can produce certain bioactive compounds like HCN, hydrocyanic acid, different kinds of antibiotic, and that can, particular and epiphytes also produce different kinds of plant growth regulator, different kinds of hormone, and those can reduce the different kinds of biotic and abiotic stress to the plants. So considering the above advantage of these endophytes and epiphytes, we have designed one experiments with different kinds of rice cultivars. The rice cultivars are Swarna, Swarna Sabwan, Sabita, and wild rice. So these are the cultivated rice varieties, the Swarna, Swarna Sabwan, Sabita. And this is, it's a wild rice that is Oryza ichengiri. This one is one African strain. The difference between this cultivated and wild strain are, during cultivation, during selection, this wild rice has lost many features. Those are very crucial to attract different groups of microbiome. Basically, this wild rice attracts diverse group of microbiome, but where this here in the cultivated rice, some of the group are privileged. Among this cultivated rice, Swarna and Swarna Sabwan, these are nitrogen responsive variety. If you remember my experiment, I'm going to discuss about nitrogen fixers. So these two varieties are generally nitrogen responsive, means if you are providing excess nitrogen than that of the recommended dose of fertilizer, you will see visualized changes in the physiology of the plant. Whereas in the Sabita variety, you will not, that the extra physiological appearance due to use of extra nitrogenous fertilizer, it's not observed that much. So we have considered these four diverse group of rice genotypes to do our further experiment. So what we did, we generally adapted culture independent approach and culture dependent approach to do the microbiome analysis. Today I'm not going to discuss about the culture independent technique, but I'll definitely discuss about the culture dependent approach. So basically what we are doing, we are harnessing the microbiome from different parts like root, stem, and leaf. It's a very complex microbiome. We are screening the microbiome based on different features, different plant growth promoting attributes, different compatibility assays, and we are making them simplified. Now we are just formulating one personalized microbiome for that particular genus or the genotype, and we are reapplying those microbiome onto these different kinds of rice genotypes. So what we did, from the culture dependent approach, we have isolated 35 nitrogen fixing bacteria from root, stem, leaf, phylosphere and phyloplane, and we have identified these bacteria based on partial polyphasic taxonomic approach. Earlier, talking with Shinjini madam, she told me that student will be really that interested if you discuss about something with phylosphere biology. These red arrow, these bacteria like Klebsiella, Pantua, and Burkholderia, these are basically isolated from phylosphere and phyloplane. So phylosphere is the region which is loosely associated on the surface, the region of a particular leaf, and phyloplane is the region on the root surface, the plane of the root. So the isolation of phylosphere microorganisms, to isolate those microorganisms, you need to prepare one buffer solution, and you need to put the leaves. You need to clean the surface of the leaves with tap water. After that, with sterile distilled water, and we need to put the leaves on the particular solution, that is the buffer solution, and you need to shake it down for like couple of hours, and after that you can do the phylosphere microorganisms analysis with that buffer, that those loosely associated bacteria, they will come to that buffer, and from that particular phylosphere solution, you need to take up the plant parts, the plant leaf, and here to isolate the bacteria from the phyloplane, you need to keep in different kinds of medium. These are generally enrichment culture. So for 2 hours onto this medium, so the bacteria which is tightly associated on the phyloplane, upon getting this much of nutrients, they will come up, and after 2 hours you need to take out that particular leaf, and from there you can grow the plate around 36 to 48 hours, and after that you can isolate those bacteria from this phylosphere and phyloplane. So after isolating those bacteria, we did different kinds of assay. That I am talking about the nitrogen fixing organisms. So nitrogen fixing organisms, the characteristics are that all these bacteria are acetylene reduction test positive. So this is the test by which you can know whether they are capable of fixing the atmospheric nitrogen or not. And another feature of all these bacteria is that they produce ammonia, extracellular ammonia. So what we did a metabolome analysis of these bacteria based on 71 carbon source utilization pattern and 23 chemical utilization pattern. And what we found that these, you can see the strains isolated from a single rice genotype, they have clustered together. So we can say that the microbial isolates, the bacterial isolates which is isolated from a single rice genotype, they have a similar carbon source utilization pattern. And here you can see one microbial isolate, bacterial isolate, that is Klebsiella. One strain is isolated from the rice variety Swarna, and another strain, another Klebsiella strain, that has been isolated from the rice variety Wild. So these strains are genetically similar, but they are metabolically different because they are isolated from different rice varieties, and they are having different amount of nutritional profile at their different lifespan. Next, after taking all those microorganisms, the objective of that project was to use all those microorganisms in the field condition. So in field, several autochthonous microbes are there. They are already there. So what we are doing, we are just preparing one microbial consortia. Before putting any kind of consortia, you need to know whether these groups of microbes are compatible with each other or not. Here you can see, say, one bacteria, that other bacteria. Suppose I am formulating one microbiome consortia with this group of microbiome. So you can see this has already been inhibited. suppose this one or this one, they are producing some chemicals or some antimetabolite that is inhibiting the growth of this microbe, this bacteria. So this permutation combination of microbes is not suitable, so they are incompatible to each other. So if you see this one, so look, see here, no inhibition is there, so all the microbiomes are compatible with each other. So before going to the field test or laboratory test with a microbiome, one need to assay the compatibility assay, whether these microbiome are compatible with each other, whether they can survive or not. And after that, the competition of autochthonous microbes of that particular niche is already there. So after taking all those microbes, we have just inoculated those microbes with different rice seedling in laboratory condition, greenhouse condition, and after that we take it out in the field condition. Here you can see this one is the control treatment, means this, here no bacteria has been inoculated with the rice seedling, and in these two treatment different microbiome has been inoculated in these two treatment. And this picture was taken after 14 days of inoculating the bacteria to the seedlings. So you can see substantial amount of growth has already been taken on the microbiome inoculated seedlings. Apart from this, we have taken this, the culture, the positive result in the greenhouse. So here you can see this one, these are the different treatments. So here it's the control one. Here nothing has been used. In this treatment, 10 to the power 8 cells of endophytic bacteria has been treated with the rice plants. Here 10 to the power 6 cells of that endophytic bacteria has been used in this treatment. In the next treatment, this one is the recommended dose of fertilizer. No bacteria, here no bacteria has been inoculated. So each rice genotype has a particular dose of recommended dose of fertilizer, that is nitrogen, phosphorus and potassium. I will say NPK. So this is a recommended dose of fertilizer, and in this treatment we have reduced the amount of urea, that is the nitrogen fertilizer, by 50%. Additionally, we have used full dose of potassium and phosphorus, plus here we have used 10 to the power 8 cells of endophytic bacteria. This treatment and this treatment are similar with the dose of fertilizer. Here we have used one bacteria at the concentration of 10 to the power 8 cells per ml, and that is rhizospheric bacteria, rhizospheric nitrogen fixer. It's not the endophyte. And in this treatment, we have used half dose of nitrogen, full dose of potassium, 10 to the power 8 cells of bacteria, and no phosphorus, because this nitrogen fixing endophytic bacteria has also the capability to solubilize phosphate. So in this treatment, we would like to see, we wanted to see whether without phosphate how it will perform. So from the subsequent experiment, we found out that at the greenhouse level, it can reduce the utilization of around 50% of nitrogenous fertilizer at greenhouse level. And at flowering stage, you can see this treatment and this treatment, it looks good. But the problem is when it entered into the grain filling stage, the number of chaps increase much more in this treatment. Chaps means the grains are there, but they are not filled up grain, they are empty grains. So there is a relation between source and sink during grain filling stage. So at that particular time, most probably the phosphate plays a very crucial role. So this treatment, we are getting much more chaps. So when we are talking about endophytes, it's very important to know the time of inoculation and when and how long you need to inoculate that particular seedling with a bacteria. We have developed one fluorescent resonance energy transfer based tracking of the bacteria, and this picture was taken after 6 hours of inoculation of a bacteria. Here you can see the bacteria has already started to colonizing on the root surface, and this is the enlarged picture of this, and you can see that they have already attached on the root surface, and this is the 100x magnification picture, and here you can see some of them has already entered into the root system. So the earlier picture, it was fluorescence microscopic picture, and this picture was a scanning electron microscopic picture. Earlier the picture was taken 6 hours after inoculation, and this picture was taken after 21 days of inoculation. Here you can see the pattern of the colonization was observed, that these bacteria, these endophytic bacteria, first they are forming one biofilm on the surface, on the junction of the main root and lateral root. When we enhance that, you can see the biofilm formation at the junction of the main root and lateral root. And here, if you magnify it further, you can see they have successfully colonized on the surface of that particular root. And if you magnify it further, you can see certain cracks, they are present on the root surface. Probably these endophytic bacteria, they are entering from the cracks of the root, and they are going to the, from root they are going to the stem and the leaf. And this biofilm formation is very much essential for formation of nitrogen fixation, because nitrogen fixation occurs generally, the nitrogenous activity occurs in anaerobic condition, so it acts as an external nodule like thing, so it will reduce the oxygen tension. Simultaneously, it will help to attach those bacteria on the root surface, and additionally it will work as a launching pad of the endophytic bacteria to enter into the root system. So this endophytic bacteria, they are entering into the root system. So if anything is invading into the plant, the question will come whether the relationship is symbiotic or it's pathogenic relation. To determine its symbiotic or pathogenic nature, we have extracted the proteome of the rice seedling, and we did experiment based on LC-MS. So this one is uninoculated plant protein, and this one is the bacteria challenged plant protein. So we found that when we are inoculating the beneficial endophytic bacteria in the rice seedling, the plant stress related protein and defense related protein are much more from the control treatment. Additionally, when we have compared our data with bacteria challenge plant protein with another pathogen challenge plant protein, we found out that proteasome mediated protein degradation, different phytoalexins machineries, pathogenicity related protein, histone modification, ROS generation system, they are very less in bacterial challenge plant protein. However, their presence is evident in when the plant is challenged with the pathogen one. Means the when pathogen is infecting one plant, this protein will go up, but we have not got the expression of this protein in our bacteria challenge plant protein. So from here we can say that plant is activating their immune system, they are actively monitoring the whole system, but the consequence like proteasome mediated protein degradation, histone modification, those are present, those are happen during the pathogen invasion, is not present in our case. That's why we are proposing it as the relation as a symbiote, it's not an opportunistic pathogen. So next, I will discuss a little bit about the bacterial bioremediation and how microbes are helping different plant genotypes to aiding that phytostabilization process. So all of us know there are several kinds of bioremediation techniques are there, like bioleaching, biotransformation, bioaccumulation, biomineralization, bioabsorption, and phytostabilization. I will show you some experimental evidence that we are performing in bioaccumulation, bioabsorption, and phytostabilization. So what is bioaccumulation? Earlier I discussed that currently farmers are suffering from a problem that different kinds of heavy and transition metal, they are coming into the farmers field from different anthropogenic activities and industrialization. So what microbes can do, microbes can simply convert those heavy and transition metal from toxic form to lesser toxic form, and they can accumulate those metal into their system so that the exposure of those metals in the surrounding atmosphere is very less. Additionally, in bioabsorption, different kinds of microbes, different algae, different bacteria, they can produce extracellular polysaccharide. So this polysaccharide act as a absorbing material of different heavy and transition metal. So one can use live as well as dead cell in the process of bioabsorption. Similarly, with the taking advantage of bioabsorption, we have developed one system that is called the phytostabilization. So here we are taking the microbes, basically those are able to produce extracellular polysaccharide. What we are doing, we are just inoculating those microbes into the plant system, and they are forming a layer of biofilm on the surface of the root, and the metal earlier which was translocated from the root to different parts of the plant, they are generally arrest on the surface of the root. So basically the exopolysaccharide produced from this bacteria is inhibiting the translocation of metal in different plant parts. So with taking advantage of that system, we tested this system in groundnut. So what we did, we have isolated two bacterium, that is one bacillus and another is halobacillus, and when we are treating those bacteria with plant, when the plants are grown on salt and metal contaminated soils, these bacteria produces two beautiful color pigments. The more we are giving the stress, the amount of production of these pigments are much more. So what we did, we simply extracted the pigment and did several antioxidant tests like DNA nicking assay, DPPH activity, FRAPE assay, and simultaneously we have identified those pigments based on GCMS and FTIR spectra, and found out that it contains substantial amount of antioxidant property, and those antioxidant property might be responsible for reduction of the stress that is generally you can found in the metal treated treatments. Here you can see these are the plant root, they have been treated with only metal. So when we are treating the metal treated plants with different bacterial strains, the damage of cell cytoskeleton is very less. Additionally, when we have estimated the metal translocated in the system with atomic absorption spectroscopy, we found that the metal absorption in the seedling a little bit lesser when we are treating the plants with the bacteria, whereas only metal plants contain little bit of higher amount of metal inside the seedling. So from here we can say this phytostabilization process is inhibiting little bit of metal translocation into that particular plant system. So simultaneously what we are doing, we are also developing this system with different kinds of algae. So this is a microbial interaction, it's called proto cooperation. What is proto cooperation? Here we are formulating that particular microbial consortia with algae and bacteria. So here algae and bacteria, they are both helping each other. How? This algae, it's a photosynthetic algae, and most of the bacteria that we are using, it's a heterotrophic bacteria. So algae is producing the oxygen into that particular system. Bacteria is taking that oxygen, growing and producing different kinds of nitrogenous compounds, and that nitrogenous compounds is utilized by the algae. Simultaneously, one of the algae, like we are currently working on two systems. One is chlorella and another is synodesmus. So this algae has also the ability to take these metals inside their system. So this is basically the bioaccumulation. So these are the control treatment, and this is one transmission electron microscopic picture which is coupled with EDS map, and these treatment are with the metal treated. So here you can see the metal, the lead has already been accumulated inside the system, and with the EDS map you can simply know where at that particular position that metal is getting accumulated. Subsequently, so earlier it was bioaccumulation. So in synodesmus, it's another algae. So these pictures are the control one, and here these treatments are the treated one, the cadmium treated one. So earlier it was bioaccumulation, here you can see bioabsorption. If you remember, earlier I have discussed these microbes can produce different kinds of extracellular material on their surface. So the green ones, the green dot one, these are the accumulation of cadmium. So what they are doing, they are simply bioabsorbing the cadmium present on that particular medium, and they are reducing the load of total cadmium on that particular solution. And my third topic of discussion, it will be on development of a biopesticide. Basically, these that was done on the tea atmosphere. So in Jalpaiguri and Darjeeling district, several tea gardens are there. So this tea garden are basically infested with different groups of loopers. They generally used to eat the leaves, the tea leaves, within couple of hours. The total leaves they will eat, and it will be totally empty leaves, and that contains huge amount of losses to the tea industries. Simultaneously, tea industry they used to use different kinds of chemical pesticide to control these loopers. So what we did, we have isolated one bacterium, that is the latter we came to know that is one Bacillus thuringiensis species that was isolated from the tea atmosphere, and we have identified that bacterium from by partial polyphasic taxonomy. And with the microscope you can see these are the characteristics feature, like they have formed the endospore, and we did several scanning electron microscopic assays. So this is the spore. So when we enlarge this picture, you can see this is the spore, and the spore is attached with the characteristics toxin, the Bt toxin, which is responsible for killing the pest. And here in this picture we have purified the spore, we have purified the crystals, Bt crystals. So this this is the spore and this is the crystal, and here they are the purified Bt crystals for further experiments. And we did different kinds of mass spec experiments with mass spectrophotometric experiment to identify different characteristics of this toxins. So here I'm not going to discuss about the molecular part, but I will discuss about the phylospheric application part. So here you can see these are the insects. So first three pictures, they depict that active feeding stage. So these are the loopers, they used to eat the plant leaf, the leaf which is responsible for production of the tea. So within like couple of hours they will eat the whole leaf. So what we did, we have formulated that particular Bacillus thuringiensis strain. We have optimized in large scale fermenter process, and here we have sprayed those Bacillus thuringiensis spore on the surface of the leaf. And these are the observations, simultaneous observation that we found. This is the 12-hour observation. After spraying 12 hours, we can see that they have stopped feeding, but the larva was alive. On 12 hours, they were alive, but they have stopped feeding. And after subsequent observations like 24 hours, 48 hours, they started to die. And here you can see they have already died, and the color of the looper has already been changed, and the next putrefaction has already been started due to congestion of the Bt toxin at 96 hours. In my title, if you remember, I have mentioned the word indigenous, that genotype specific microbiome, that is personalized microbiome. That benefit of personalized microbiome is that these looper belongs from the family of Lepidoptera. If you go to any kind of tea garden, you will see different kinds of insect, indigenous autochthonous insect present on those field, on that specific niche. So they belong from different group of like Lepidoptera, Diptera, different families. It has been reported from different papers that Bt has their effect on other families too, like Diptera. So from this indigenous species, from the species which has been isolated from that garden, we found that there is no effect on the beneficial insect that are present on that specific niche, but selectively it's killing the tea loopers which are belongs from the Lepidoptera family. So in conclusion, we can say that the diversity of endophytic and epiphytic diazotrope in different rice parts, different plant parts of rice has efficiency to the niche, as in different rice phases, like in reproductive phase, in flowering phase, in pre-flowering phase, the nutrition is different. That's why the microbial dynamics used to vary all these stages. We did all the experiment at pre-flowering stage, because at the pre-flowering stage is the most active phase of any kind of plant genotype. Subsequently, microbiome exhibited better vegetative and growth in pot and field experiment. This AB2 strain after that from the lab experiment, where it is able to like reduce the amount of urea 50%. So when you take up in the field condition, it can reduce the use of ammonia around 30%. And during last 7 years, it has been tested in different parts of India, and this bacteria is able to successfully colonize at the root of the plant. Next, the plant microprotocooperation can reduce the saline and metal stress in agricultural field. So what we are adopting, this called the phytostabilization process. Basically, they are reducing the translocation of the metal due to formation of the biofilm on the root surface. So basically, that biofilm is inhibiting the translocation of the metal. And the lastly, indigenous polyvalent microbes should be considered to control the pest. Like indigenous, it should not compete with the autochthonous microbes. Otherwise, in a specific niche, if you are, suppose you are isolating one bacteria from a different state, and you are implementing that specific bacteria in, suppose, West Bengal, you have isolated one bacteria from Kashmir, you are implementing that bacteria in West Bengal, the total microbiome is totally different. So the indigenous, it has been seen that indigenous microbiome, they generally outcompete the outsider. So if you are using this indigenous one, the chances of outcompeting the inoculated microbiome is very less. And polyvalent means it's not only it will have the biocontrol activity, we are using that particular bacteria in phyllosphere and phylloplane. So if it has some kind of activity, it can induces the plant growth, that will be fantastic. I'd like to thank Dr. Shima Banerjee, principal of Hooghly Women's College, for inviting me for the talk. And I would also like to thank all the faculties of a microbiology department, Dr. Piali Guho, Mrs. Doel Roy Nag, Mrs. Shinjini Dhor Haldar, for inviting me and giving me opportunity to talk about my research. I would also like to acknowledge my mentors, Dr. T.K. Dangar from National Rice Research Institute, Professor Subhrakanti Mukherjee, Department of Microbiology, Bardwan University, Dr. Upendra Kumar from National Rice Research Institute, Professor Edward Blumwald from University of California, Davis. And I would also like to acknowledge Professor Devashish Das and Dr. Sanchita Goswami. They are synthesizing the ligands for me to do all the tracking. Additionally, I would like to also acknowledge my student Chandrasekhar, Rutvika, Pooja, and Maya. Chandrasekhar, he is working on the algae system, algae bacterial consortia system, and Rutvika, Pooja, and Maya helped me a lot to develop the peanut system. Thank you. Now the session is open for the questions. I think I have completed within time. Or I'm going ahead. Yeah.
Okay, now questions. We have sorted out some questions. Now we are putting it.
Okay.
First question is, how a layman can differentiate between symbiotic and pathogenic strain?
Basically, they have to wait for the phenotype. Layman, it's like phenotypic changes will be there. So for scientists, it's late. It's too late. That's why we can adapt different kinds of molecular biological technique, as well as different kinds of. I'm stop presenting. Different kinds of like volatile. We can estimate the volatile. The total signature will be changed. But for layman, they have to wait for the phenotype of the particular plant. They will see for cropping plants, production will be less, and the chlorophyll content, the physiology of the plant, it will be like the physiology will be not good. Physiological condition of the plants will be not good.
Okay, the next question, how to standardize the number of cells per ml?
Standardized Bureau of Indian Standard, they have already one recommended dose. So here I have not shown all those things. You have to inoculate the plant with different doses of microbiome. So the lesser dose is effective. Like in my experiment, you can see I have here demonstrated two loads. One is 10 to the power 8 and another is 10 to the power 6. So 10 to the power 8 is much more. And Bureau of Indian Standard, they have certain guidelines. You need to follow that guideline, and the lesser amount of that particular load doing maximum amount of plant growth promotion, you need to take up that particular concentration.
Okay, thank you. The third question is, how stress and pigment production are related? Can you explain it again? Participants have written that. You have already explained, but they want the explanation again.
Actually, it's a like of Ross generation, reactive oxygen species. So when stress is there, plants generally produce different kinds of Ross, reactive oxygen species, and this pigments has the ability to reduce the Ross, the effect of the Ross. If you remember, we did assay it for several antioxidant properties. So these antioxidant properties, the advantage of using microbes is that it was evident that when we are increasing the stress, maybe the bacteria is trying to reduce the stress in its system by producing the pigments, so that it can reduce the stress in its system. Simultaneously, it's producing the pigment, and that pigment can be utilized for the plant to scavenge the reactive oxygen species.
Okay, thank you. The fourth question is, what is Phytostabilization.
Phytostabilization I have already discussed. Like it's stabilizing the translocation of the metal inside the plant root. Basically, for that, what we are doing, we are selecting the microbes which can form the biofilm. So they are forming the biofilm on the surface of the root. So at that particular portion, it works as a seed. So the metal cannot pass through that seed, and they are getting arrest on the surface of the root, and they are not getting translocated into that particular system. And thus the effect, the metal accumulation is very less. So that's called the phytostabilization. It's stabilizing the effect of the metal on its surface.
Now the fifth question. What are the extracellular products and how do they make impact?
Extracellular, I think he or she is asking about the polysaccharides. Is it? I think it's a matrix.
This
It's
is
a,
mentioned actually.
yeah, basically I got what he or she was asking. Basically these are extracellular polysaccharides. If you are microbiologist, you know, they produce different kinds of polysaccharides, glycocalyx, slime layer. These are made up of different kinds of polysaccharide. It's a biofilm, the composition of the biofilm. So these are act as a attachment on the root surface. So it's a attaching material, and that it can, like, different bacteria can colonize over there, and it acts as a attachment of the beneficial microbes on the surface of the root. So these are basically the polysaccharides, different kinds of polysaccharides.
Okay. Thank you, sir. And there is no more questions. So thank you so much for your wonderful explanation about all these things, and it was very encouraging for the students of microbiology because it was also related to their topic. I hope they find it very interesting
I
and
hope
fruitful
so.
to
Yeah.
their syllabus. Okay,
Yeah.
thank you so much, sir. Now over
Yeah.
to Sinjini.
Thank you. Thank you for giving me the opportunity.
Thank you, Dolby. Now, our next speaker is Dr. Devushmita Mukhopadhyay, and she is an experienced molecular biologist trained in cancer research with a therapeutic expertise, and published more than 20 research articles in reputed international journals. She has completed their master's in molecular biology from Presidency College, Kolkata, and joined for doctoral studies at Indian Institute of Chemical Technology, Department of Chemical Biology, Hyderabad in 2012. She is skilled in development of novel small molecules for their anti-cancer activities, translational research, mechanism for drug action, and understanding the molecular events and signaling pathways that are the consequences of various types of cancer and other abnormalities in order to find out potential therapeutic targets with a special emphasis on microRNA. Later, she joined her postdoctoral fellow in American University of Sharjah, UAE, and established her excellent command in the field of cancer drug delivery using nanocarriers that involves surface modification, tissue culture, biomaterials, non-viral gene delivery, and efficacy analysis of drugs in vitro and in vivo. She is also having expertise in the field of diabetes and other metabolic disorders involving microRNA and novel genes associated with hyperglycemia. Our today's topic is new trends in cancer therapeutics, time to think biologically and logically. Now, over to you, Dr. Devishmita.
Hi everyone, I'm audible.
Yes, ma'am.
Thank you, Shinjini, for your beautiful words. And before starting everything, I would like to say a very good afternoon, virtual good afternoon to everyone. And I would like to express my warm gratitude towards the organizing committee and giving me this opportunity, arranging this webinar and giving me this opportunity to be a part of this. A special thanks definitely goes to Principal Madam, uh, Dr. Seema Banerjee, and Dr. Shamulina Goswami, and of course for inviting me, Mrs. Shinjini Thor. So, as I can understand, this is a Google Meet platform. So let me just share you my presentation to you all, and please confirm whether my presentation is um, viewable to everyone or not. Is it okay? Can I start now? Hello. Okay. Okay. Hopefully this is good. It's still not visible. Okay. Let me get you the one. Now it's okay. Thank you so much for confirming. So, myself, Devashmita Mukhopadhyay, I did my PhD studies from Indian Institute of Chemical Technology from Hyderabad. And then I shifted to UAE, and presently I'm doing my postdoctoral research fellowship in cancer therapeutics and delivery technologies. So, since my PhD days, I was involved in cancer drug therapeutics. So, today I know the world is very much concerned about the one of the biggest pandemic, that is COVID-19, and its related threat to us, and eagerly waiting for some cure or vaccination. But today, definitely, I'll be presenting something a constant problem, a major health burden towards us, mostly known as cancer. So, what will we discussing today? So, a brief about the basics of cancer, causes and characteristics, becoming a global threat day by day, present types of treatments which are available, and more approach towards targeted therapy over chemotherapy, a personalized medicinal approach. And then I'll discuss a little bit about my present two projects, that is ultrasound mediated liposomal drug delivery, and another project is small molecule modifiers of microRNA, or you can say is a non-coding RNA as a gene therapy. And definitely I will proceed towards questions. So here you go. So basically cancer is, as you all know, that is a global burden now and it's becoming a major. But in day by day, each year, tons of millions of people are diagnosed with cancer, and more surprisingly, that half of the patients eventually die for it from it. So basically, what is cancer? It's a disease caused by an uncontrolled division of abnormal cells, and the most common part is that the malignant growth, that is a metastasis of the cancer when it is happening inside the body, and that is the most dangerous part, that it is uncontrollable. Even a single chemotherapeutic agent followed by a radiotherapy, it cannot stop a growth of the cancer, and eventually the patient is die. So, what's the causes of cancer? So, as you can see, there's a various reasons behind it. That cancer, it's biological and internal factors are involved: age, gender, genetic defects, different environmental exposures, skin types, definitely occupational risk factors, including carcinogens, chemicals, even our recent diet protocol, and present lifestyle-related factors are also involved. So, what are the characteristics of it? As you all know, starting from the immune destruction to replicate immortality, invasion and metastasis, activation, angiogenesis, induction, resistance of cell death, and sustaining proliferating signaling, evade growth suppressor, progress genome instability and nutrition. Everything is associated with cancer. So totally, it's a total destruction of our immune system and every body, including the you can say the multi-mode role party, is actually getting affected by cancer. So here is a brief about our global threat. Why it is a global threat? As you can see that day by day the world is getting engrossed with the cancer threat. And if you see the male-female versus scenario, still the lung and bronchial cancer is the most dangerous and leading the pathways in cancer death. While in male, the next most dangerous is the prostate cancer, and in female, it is ruling out the breast cancer. So now I'll come to my exact research protocol that types of treatments of cancer nowadays is available. First of all, the surgical intervention, and definitely it is the you can say when there is no option late, then the surgical intervention has come. Followed by one chemotherapeutic approach, radiation approach. But nowadays, it is not about that you can say the treating the cancer at the late stage. It's about the diagnosing the cancer at the early stage and providing the basic treatment against it. There comes a more personalized therapy, the 4, 5, and 6, the gene therapy, immunotherapy, stem cell, and bone marrow transplant. So obviously we are now approaching towards a personalized medicine, and we are approaching towards a targeted therapy. While chemotherapy is it is very much you can say chemotherapy is useful, but it has brings a lot of side effects. Where the targeted therapy is inhibit a more specific target in cells, and many are even oral agents. It's a mixture of cytostatic and cytotoxic materials, but it is more precise to a particular cell, which are known as a cancer or tumor cell, without reaching any hamper to the normal healthy cells. And there is an advantage of the targeted therapy or personalized therapy comes into it. So here I have discussed, I will be discussing about the personalized medicine, think biologically, right? Till today, there's a like we are giving some chemotherapeutic approach to everyone. Like someone is coming with the breast cancer or prostate cancer or whatever, we are giving some chemotherapeutic approach. It might not affect to the particular patient because his or her immune systems are different. But nowadays, what is the future medicine is coming along? It is an approach to patient care that allows doctors to select treatments that are most likely help patients based on genetic understanding of their disease. This is a very important term, genetic understanding, because there actually the evolution comes. And the very basic of personalized medicine comes into the picture. So if you can see, there's a one particular motif is that with the gene therapy or small non-coding RNA or microRNA therapy. Basically, what does it do? It actually degrade the protein technologies to target the undruggable oncoprotein. There are some cells which may become some metastasized or advanced stages of cancer has already been happened. So now the exact chemotherapy, which is nothing but a chemical drug, right? And it is not, it is beyond its action. It cannot actually regulate the overgrowth of the cell. Here comes our own immune system, our own genes. Nowadays, our personalized medicines is working along with these genes and immune therapy so that without taking any you can say the outside medicine or any chemicals from outside, we are actually inverting our own immune system, our own genetic implant, so that we can fight against cancer. And this is the motto behind the personalized medicine, to use your own immune system against cancer or to fight back cancer. So part one of my work is presently we are doing here. This is my university right now, American University of Sharjah, and this is my lab. It is saying the ultrasound mediated liposomal drug delivery, a focused drug delivery approach. So even if we are talking about chemotherapy, are our drugs actually going to the specific cells? Maximum cases answer is no. Because if you are taking, if any patient is taking, say for 100 mg, 200 mg of drugs. Only 10 mg is going to the specific tumor tissues. And top of that, the other chemotherapeutic drugs are actually killing the healthy cells. Whereas our current approach is the drugs that are encapsulated inside the targeted nanocarriers so that after getting injection, the targeted liposomes can accumulate in the tumor site without affecting the healthy tissues. Ultrasound waves and then applied to release of the drug from the liposome. So, what are nanocarriers? Like a plethora of chemistry work has been done, and me especially being a biologist, even understand not even 5% of this nanocarrier work. To be very frank with you, there's a chemistry team working day and night to developing most possible nanocarriers and challenging nanocarriers. Those can actually extravagate the cellular cell membrane and reach towards uh, exact tumor tissues or malignant tissues. So these are nanocarriers actually being used to a transport module for another substance such as drug. Commonly, there are multiple nanocarriers available. There is a polymeric carrier, polymeric micelles, dendrimers, liposomes, solid lipid, cold carrier, even viral carrier. So what are the mode of action of these nanocarriers? The drug molecules carried by nanoparticles are released in the extracellular matrix and diffuse throughout the tumor tissue. The particles carry surface ligand to facilitate active targeting and receptors present inside or on the target cell of the tissue. And what are the advantage of it? The most technological advantage of nanoparticle is as a drug carriers are highly stable and high carrier capacity. Feasibility for incorporation of both hydrophobic and hydrophilic substances, and feasibility of various route of administration. The large surface area has also a large affinity for drugs and small molecules like ligands or antibodies for targeting controlled release purposes. Some cells which are maybe they do not need, they are not in a malignant state, they are about to start the malignancy. So they actually don't need the 100 mg of the drug. So here comes the controlled release of liposome. The particular liposomal drug will achieve at least 10 to 20% of release for against those particular cells which do not need maybe 80% of the drug. So that's why it can actually avoid uh, the mortality of the healthy cells. So presently, my lab actually focused on the liposome mediated drug delivery. So what we are doing here, we are different anti-cancer drugs are there, like herceptin, like doxorubicin. These are all potential cancer drugs which are established. And what we are doing right now, we are doing a liposome having a core of hydrophobic and hydrophilic. So we are incorporating doxorubicin inside liposome and inside pegylated liposomes actually, or we are targeting this liposome by attaching some receptor molecule. Say for breast cancer is having some specific receptor moieties, so EGFR, Herceptin, these are some breast cancer specific receptors. Now, I want my liposomal drug to sit and deliver doxorubicin only specific to this breast tissue. So this liposome come into this picture, which are having tagged with the anti-receptor molecule on the surface of it. As you can see, this receptor molecule, the herceptin, EGFR, this molecule are on the surface, and doxorubicin is the inside of this liposome. So what it will do? It will basically recognize the breast cancer because of its surface receptors moieties, and it will sit on the breast cancer tissues only. After sitting into it, it is known as an address point for them. They only will address the breast cancer tissues first, and after addressing these tissues, they will sit there and release their internal drug, that is doxorubicin. So what we are achieving here now, we are achieving most targeted and specific release of our drug. That is the whole purpose of um, nanomedicines or nanocarrier mediated medicine. So, I'm sorry. So, what is now? we normally produce a liposome moiety and then we cross check what is our delivery potential against a normal cell. Here we actually use a flow cytometry machine that estrone is another receptor conjugated liposome, showed higher uptake by the estrone positive breast cancer cells compared to estrone negative breast cancer cells. So the blue peak here is basically estrone negative breast cancer cells and the red peak is the estrone positive breast cancer cells. And the main motto is to supply this particular drug or liposome inside that red zone. And as you can see from the picture, this estrone positive breast cancer cells actually having estrone receptors on it. So this liposome actually can recognize the breast cancer positive cells and feed onto it, then release the drug showing the higher fluorescence, uh, uh, fluorescence activity here. So flow cytometry analysis here is showing the blue color breast cancer cells, whether it is not getting the much liposomes, where the estrogen positive breast cancer cells, which is the red color, is getting focused delivery of liposome here. But the main problem is that liposome sometimes cannot cross the cell membrane because as tumor microenvironment is very complex. Somewhere the tumor microenvironment like the cellular membrane is very rigid sometimes. So we are expecting that liposome will go through inside the membrane, but actually practically it is not happening. There we need some triggering agent. And our lab has been specialized triggering agent has been used ultrasound as a special triggering agent. Because ultrasound till date, it was actually mechanical wave about human hearing ability of 6 kilohertz and it was a cyclic sinusoidal acoustic wave. But till date, it was used for diagnostic purposes to study a developing fetus or a medical imaging purpose, a person's abdominal and pelvic organs, muscles or tendons, and their heartbeat and blood vessels. But it can also do perform as a triggering agent, and this work has been carried on in our labs, and also some other parts of the lab, definitely part of the world. In triggered drug delivery system, ultrasound helps to increase cell permeability by disruption of the liposomal bilayer and trigger drug inside the tumor microenvironment. The main aim is to find the optimum ultrasound parameters that permit enhanced drug delivery without even harming the other cells. So as in the picture you can see these are different nano carriers and it is somewhere getting uh, it's not effective in crossing the cell permeability, whereas the sonofuration come into the picture and using thermal or mechanical waves and cavitation mechanisms, low power ultrasound or high power ultrasound actually evade the cell permeability and helps those nanocarriers to go inside the cellular membrane and work accordingly. So now I am going to show you actually what work are going on in our lab and how from a book to a laboratory how this work has been performed. So as you can see, I'm just here have portrayed with a single example. We have worked on this colon cancer cell lines, and in vitro work has been done. And here I have portraying you the in vivo work, the actual original work of this ultrasound mediated liposomal therapy. So what we have done, we have taken this for nude mice. Nude mice is an immunocompromised mice, and you can actually develop tumor by inoculating cancer cells. So we injected cancer cells, which is HCT116, which is a highly aggressive colon cancer cell. We injected in the right flank of this nude mice, and this is the picture I have taken when I actually weigh the nude mice, because during the whole procedures, some nude mice, they tend to lose weight. So we cannot take them into consideration here because of the side effects of the therapy or sometimes stress, they eventually die. So 22 to 25 grams of weight is very perfect for any experiment to start with in animal model. So here I have shown you that this nude mice model I have taken and inoculated tumor in the right flank lesion. Tumor normally appears after 10 to 12 days, around 100 mm cube. So this is a small tumor when actually you cannot actually wait for when the tumor will go big, because that time your other cells have been affected. The you can say the metastasis also has started. So definitely you have to keep a very close eye on this mice every day to check whether there is a small tumor appearing or not. And this is a small tumor which is appearing after 10 to 12 days. We have observed it is a very small spot, but a very hard spot, which is known as a tumor here. So now all node mice have been divided according to our experimental protocol. And what we have done here, we are treated the liposome treatment intravenously. Actually, the exact picture I was not able to take during our intravenous injection, so I have given here a demo picture. Why we portraying is an intravenous is also called a tail vein injection, because the node mice is very delicate. It's very small. You can, if you have seen it, you can understand it's very small and it's hard to get a possible vein in his hands or leg. So it's the largest vein is there in the tail portion of the nude mice. So we have given the intravenous injection using tail vein, and the total amount of liposomal drug was 8 mg dox per kg of the body weight. And followed by that, we have performed some ultrasound 5 minutes prior injection and 8 minutes post injection in every alternate day for 8 days. Okay, so here I will actually show you how it looks like. So yeah, if you can are able to see, this is called sonoporator. The mice here has been given some sedatives. So because we have to apply ultrasound for 8 minutes or 15 minutes at a time. So this is sonoporator we have applied on the mice of this particular tumor, where the tumor is there. We cannot apply to the other skin portion, otherwise it will burn the skin. So before application, what we have done, we have subjected to some gel, ultrasound gel, and then using this sonoporator and exact parameters, we applied ultrasound on this mice. So as you can see, this is like a gentle massage on the tumor. Yes. And we have to apply it for continuous 8 minutes. So I have just taken a clip of it to share you the views. Yesterday only we have performed it. It's called the ultrasound treatment on the tumor, mice tumor. Okay. So, sorry. I think you're there. So then we applied ultrasound just to show you. And meanwhile, the control group, which is not having any treatment, it is growing. The tumor size is growing a lot because it's ultimately it is not getting any treatment. So at the end of 28 days, like 28 days, why the protocol has been performed, because we have to observe that what is going after completion of our whole treatment, even the injection followed by ultrasound, we cannot just take out the mice, because we have to observe them how whether they are getting weak, any other side effects are coming, what are the other effects that are actually harmful for the mice or not. So after 28 days, we are actually taken out all the mice group-wise and we have sacrificed them. As we can see in this picture that there's a different tumor sizes are there, and this is the control, the biggest tumor size, as you can see. Even this is like some point of time we can see that our ultrasound didn't even work. So in this point of view also we can see the biggest tumor, but as you can see, this is an experiment, anything can happen here. So this picture is just to show you that after 28 days when the mice model has been sacrificed, the all tumors look like this. Finally, we have to take out the tumor and other organs also and actually measure by slide calipers. Here I have taken a scale just to show you that how small they are in sizes. This control tumor is obviously the biggest one. And we have got that doxorubicin very the tumor size were very less, but we couldn't get a matured result using ultrasound. And these are some our other experimental protocols we are still working on. Okay, so what next? After getting the tumor, now we have to do some biological process and do the HPLC analysis. To get what is the HPLC analysis will tell us. This will actually tell the tox content, that is our drug content, inside the tumor and other organs. So what is the fundamental rule behind it? We need to get maximum drug inside the tumor, whereas the other organs should not receive much drug. Now I'm going to show you this is a failure result here, because here we have not got enough reduction of our um, like doxorubicin. Yeah, definitely it's less than the control, but we are expecting much more better result. This is the doxorubicin content in tumor. That means the ultrasound actually helps um, the, the liposome, the doxorubicin content liposome, to invade cell permeability and go inside the tumor tissues and start their activity. Okay, though the result is not very promising, because we are now standardizing the protocol, and definitely this method is going to work. Whereas this heart, dox content in heart is showing is almost similar in every aspect. Like control is wherever heart is control heart is getting as much doxorubicin as normal other heart content of the other experimental mice, even after applying ultrasound. So the next part, I'll be sharing you that the first part will told you about the delivery technique, focused delivery technique, where the part two with the gene therapy, which is my part of my PhD work, which has been done in CSI Indian Institute of Chemical Technology. It is a work with small non-coding RNA or microRNA as cancer therapy. So, as you all know, that microRNA is a new class of regulatory molecule, and it's very important because it's a role in RNA interference. It's a biological process with small non-coding RNA negatively regulate gene expression. I just shared you my view on this, but in spite of getting some extracellular things, some chemical out from outside, it's obviously better to get ready our own immune system, our own genetic implants, so that we can fight against cancer, right? So this approach is one of them. RNA interference is microRNA, something which is present inside our body, and it is very much versatile and as well as constant and conserved in higher to lower eukaryotes. It can modulate gene expression and control cellular timing. And recent estimations support even to that more than 60% of the human coding genome is directly regulated by microRNA. So it has role on cell proliferation, cell differentiation, development, epigenetics, insulin secretion, apoptosis, stem cell, blood cell, every aspect. Okay. But when we are talking about small molecule and microRNA inhibitors, how does it work? So there is some oncogenic microRNA inside the body as well as the tumor suppressor microRNA inside the body. So oncogenic microRNA is something is not needed. So there are small molecules. In my research, I have concentrated on the natural product, which I'll explain in later slides. So I have concentrated on the natural products which actually can inhibit the oncogenic microRNA inside the body, as well as it can trigger up the tumor suppressor microRNA within the body at the same time. And what the after effects of it? Now I'll explain you here. So when we take some microRNA as well as some small molecule, how does it go in the lab? So I have prepared these slides keeping in mind the junior students in the webinar who is attending there. So for them, it will be very easy to understand. See, cancer is a vast world and you cannot work in every aspect. So how to start the cancer project, and how will you know that what is your interest and which line of the cancer line you want to work? So you have, if you are working with the small molecule modifiers of the cancer or therapeutics, then your first step is to screen small molecules against a panel of human cancer cells. cancer cell line. Now, what are the small molecules? Small molecules are already either your institute has some biobank small molecule, or you can even take some phytochemicals or natural small molecules which are already present in the nature, or you can even go with an established anti-cancer agent, and you can go for you can even discover some new molecular therapeutics, some new molecular pathway, and invent something new. And definitely you can further propagate with us existing small molecule and even do in a better therapeutic approach, targeted therapeutic approach. To first screen small molecules against a panel of human cancer cell lines and confirm its effective dosage. Second thing, effective dosage means you have the panel of cytotoxic assay are there present. So you can follow the cytotoxicity assay and confirm whether those molecules are actually cytotoxic or potential against human cancer or not. Second is the validation of the lead molecule through other cell-based assays, because cytotoxicity is not the only thing. You have to do some other aspects like flow cytometry analysis, tunnel assay to know the DNA, RNA content of this particular cell, apoptotic assay, whether the cell is undergoing apoptosis or cellular death or not, which particular cellular cell cycle it is existing in, etc. So you have to establish and validate the lead molecules. Maybe out of 1,000 molecules, you can get only one or two molecules, but that is your either trigger point. Third is establish molecular pathway affected by the small molecule. I just told you that. Then you have to do multiple experiments using DNA, RNA, PCR analysis, Western blot analysis, even immunocytochemistry studies to boost up the exact pathway and molecular details that which pathway it is affecting it. And you have to establish. Now you're talking about science, so you have to establish the basic pathway. Now the next step is establish link between target gene and microRNA using in silico analysis. So now here the question is why microRNA? Because we can actually work on the small molecule. So here, if you can, using the small molecule, if you can activate something inside the cell internally, okay, so your dosage will be very much less compared to only chemotherapeutic. So how to do it? There is a different softwares available to give you the prediction site in your mRNA, microRNA prediction analysis. Once you know there are small molecules working on this particular XY gene, that XY gene is your target gene now, and you have to establish the link between the microRNA and the gene and confirm the effect of the small molecule on target microRNA using real-time PCR analysis. Finally, you have to establish every aspect using in vitro and in vivo experiments. Now coming to the lab picture, was that, see, this is a project I have done during my PhD time. This is something, it is known as some moieties in the azoflavone. Now, let me tell you, azoflavone is a very, very common anti-cancer therapeutic drug, and it is even naturally available in different stain and bulk of different medicinal plants. So these are very simple in structure. So what we tried to see is we picked up two different molecules, C3 and C4 actually I named it, and we did some MTT assay. MTT assay is a cytotoxic assay in different cell line to the first step, which I just told you, to actually portray whether this molecule is having any effect against this cancer cell lines or not. So MTT assay of HEC is the normal cell line, which we will also have to confirm this. Particularly, this molecule doesn't affect the normal cells. HEC is the normal cells, whether it has a tremendous cytotoxicity against when it is applied to the different cancer cells. MCS7 and MDMB cell lines are used for breast cancer cells. ACE2 is a different Drosophila cancer cell line model. So next, we have even the other cell lines work also been done. We here have not portrayed, but it has been done at least in 10 different cancer lines. And then we finalize the effect of mostly the cytotoxic effect against the breast cancer cell. So we performed this experiment, the followed experiment on the breast cancer cell lines. So next step is the flow cytotoxicity cytometry analysis, which will tell you that where it is acting. Now you can see this acting as a cytotoxic potential. but where it is acting, which cell cycle, particular which phase is getting arrested, that will be telling by tracks analysis. So here comes the microRNA. I have skipped some slides. Basically, have not portrayed into it because I thought it will be very complex for this presentation. Meanwhile, we have already analyzed that which pathway, which particular gene is getting affected by these two small molecules. And we think that it is elaborately is affecting metastatic pathway, which is the Hoxsey, Jane Rose. These are some important players. Very those who are working in cancer, it might be very well known and familiar genes to them. So it is actually working on the metastatic part. So my next work is to link with those genes with the microRNA. So now let me tell you, there's a panel of microRNA which I got from some in silico analysis. Those can be direct or indirect target of those two genes, metastatic genes, and as well as by literature search also we analyze that these microRNA are the direct player of metastasis. Some are negatively, some are positively. So we chose a total panel and tried to identify if this small molecule is working on the metastatic pathway, then where it is working, which microRNA is getting affected. As the result in front of you, as you can see that microRNA can be, which is a positive player of metastatic, is getting inhibited by this compound because this compound is acting as an anti-cancer agent, right? So the microRNA can be the cell growth is getting affected. So next is to work is to just molecular docking, because we have to scientifically prove that this molecule is actually binding to the microRNA structure. So these are some like work we have done in silico. This is a computerized program. Some softwares are there. We took help from them, and we established that molecular dock model for microRNA 10B with as a flavin derivative C3. Because out of these two molecules also we have got the C3 is much more potential than C4, so we continued with only C3. So here is just to give you a brief idea about that. How does it work? See, I told you continuously that we have to establish a multiple pathway. So these E-cadherin, vimentin, this twist one, these are all genes which are actually the key player of the metastatic pathway. Okay. So what we try to establish that microRNA, the internal medicine, you can see, it is getting inhibited by this small molecule actually, and eventually it is helping to reduce metastasis inside breast cancer. And these are all immunocytochemistry slides we have prepared, as well as some protein Western blot we have prepared to go to individual marker and prove that yes, they are getting affected, and this particular molecule as a working as well as microRNA B modifier, and also it is helping to reduce metastasis inside breast cancer cell line. Next, obviously, the xenograph mice model work we have to do. So going much into detail, I can show you this is control tumor. This is only microRNA 10B. Only microRNA 10B means it is actually nothing, because microRNA 10B is obviously it is a metastasis inducing. But when I applied my compound in microRNA 10B, see the growth is amazingly almost 60% regain. And then we also portrayed a graphical expression on it, and eventually we isolated or extracted tissues from the tumor, and we did individual safe or whatever study we have done for in vitro using cell lines, we did with the in vivo as well, and so similar kind of result we got. So I can confirm that whenever whatever hypothesis I applied using in vitro model, the similar pathway is working even in mice model system. So after doing all this study, we can now establish a pathway that this azoflavone actually inhibitor of microRNA 10B. And these are some extra work we have been done. The slides have not presented here, but we finally established that it is working as a P10 or PI3K AKT pathway reduction manner. It is also inhibiting the EGFR genes. And finally, what it is doing? It is doing it is inhibiting the the metastasis and cellular growth of cancer. That is the ultimate thing. So, why these two models? Now, I am I'm going the brief about it. So, basic approach of this presentation is we are moving. Definitely, we are moving to get a promise to reality in cancer cure. One approach, the first part of my work, to do something betterment in the cancer drug delivery. Second part is showing you to play with our own immune and genetic system and produce something logical. Okay, so that's why I given it a not. It's also logical as well as biologically progressive. So now, whatever precision medicine or personalized medicine has come into the picture for future cancer care, it is giving you not only the chemotherapeutic approach, it will give you a combined collaborative approach of integrated care, collaboration, changing skill set, early diagnosis, which is very important. Managing cost. Obviously, managing cost is the most important thing in any drug industry. Immunotherapy, genomics, and of course the data analysis, achieving the world fear of cancer. Finally, I want to acknowledge each and every member of this organizing committee, especially Dr. Seema Banerjee, Principal Hooghly Women's College, Dr. Shamlina Goswami, Head of the Department HWC, all organizing faculty members and staff of Department of Microbiology, Dr. Kiran Hazra, Technical Coordinator and HOD Department of Physics, Mr. Satyajit Viswas, Department faculty members, Dr. P.L.E. Guho, Mrs. Suil Roynav, Mrs. Shinjini Haldar, my present postdoc PI, Prof. Ghali Hussaini, who is a HOD of Cancer Drug Delivery Program at University of Sharjah, Dr. Upal Bhadra and Manika Balbhadra, they are amazing supervisor I got during my PhD sessions, which I did from Hyderabad. My beautiful colleagues, amazing lab members, and of course my family and their support. Thank you.
Thank you, madam, for this wonderful session. So we are having some questions over here. The questions are, how does ultrasound treatment control the growth of tumors and does it have any side effects?
Yes, ultrasounds have impact on cellular growth as well. It has been seen that ultrasound has a different wavelength. Okay, so when it is applied on a very low frequency, it has some different aspect. Whenever it is applied on a high frequency, it has some different aspect. Now, for imaging purpose, sorry, I'll just switch on my video. Okay. For the imaging purpose, it has been seen that when the low frequency ultrasound can help in the imaging and you can see the diagnostic purpose, whether the high frequency ultrasound will give you particular wavelength can give you uh, you can say the focused delivery pattern, which will eventually help your uh, liposome or other nanocarriers to go inside the cellular membrane. And definitely, when liposome with drugs is inside the cells, it will eventually inhibit the cellular growth, the tumor growth. So yes, ultrasound definitely a prime role has a prime role in inhibiting cellular.
Okay, our next question is what should be the minimum drug dosage that needs to be transported to tumor cells?
Well, it is definitely a very much variable. It depends on the patient to patient and obviously a state of the cancer. So when a child is getting affected, definitely her or his body weight is much less than the adult. So we have to consider the two factors here. One is the body weight. Normally, we started with the 100 mg or 200 mg based on the cancer type and the state of the cancer. Well, if it is a like very early stage, we definitely will start with a very minor or low potency, that is 50 mg or 100 mg. When it will go towards a very active state, maybe only drug will not be effective. We'll also take the immunotherapeutic strategy as well as ultrasound and started with the radioactive potential.
Okay, and the next question is, what is the future prospect of using siRNA in cancer therapy?
mRNA.
No, actually, he has just written siRNA.
SIN, okay, small interfering RNA. See, small interfering RNA is something which is, it is again the non-coding RNA, but remember one thing, it is something from external outside. So small interfering RNA is something which actually design, it's a 20 to 25 nucleotide base pairs we design accordingly to regress any particular or suppress any particular gene. So siRNA has been tremendously used in research purposes to know whether the genes, the actual role of the gene or the important purpose of the gene inside the cell. But siRNA cannot be a role model because miRNA is there inside the body. And you have to understand one thing here is the most approachable thing is to make use of our own body system. siRNA can be a good therapeutic strategy till we know the purpose of the which cell or which gene is getting involved to for the particular cancer. But once we know that this gene is absolutely okay to treat with or absolutely okay to go further or, in our cancer therapeutic, then our next strategy will be miRNA, microRNA, which is sitting inside our body, not siRNA.
Okay, and the last one is, what does the personalized medicine depend on?
Sorry, can you repeat?
What does the personalized medicine depends on?
What?
Does the personalized
What
medicine
does
depend
Okay, personalized medicine is something very important to say that it is not something only which will follow only one step. Some patient will go and say, I have this problem. So first we have to analyze that patient coming from which background and cancer, how old is his cancer or her cancer. Okay. So now we have to justify from his blood, urine sample, we have to give a genetic implantation to his total body system. So now immune system, like depending on his or her own immune system, the personalized medicine will be generated. Number one is immunotherapy. Immunotherapy consisting of not only DNA, RNA, it will also consist of the antibody, monoclonal antibody. Suppose she is suffering from breast cancer. We will now pretend that the which type of breast cancer she is suffering from. The breast cancer is now being a very elaborate term for her. Now, personalized medicine means so it will be very much personalized. Which kind of breast cancer? Herceptin positive, Herceptin negative, GFR positive. If Herceptin positive, then we have to identify which antibody is present in particularly that patient's body to generate a monoclonal antibody. So we'll generate the monoclonal antibody depending on her or his immune system and will tagged with the liposome and using ultrasound will deliver inside his or her body. So that much personality. It is not only one step or two step process. Monoclonal antibody is one aspect, gene therapy is another aspect, and most of the cases it has been seen that it is working as a total. Like we have to apply the gene therapy as well, the monoclonal antibody body therapy as well, and as a stem cell or pluripotent stem cell therapy as well. So together it will work on his or her immune system to generate NK cells so that he or he can fight back the cancer.
on? Okay. I think we don't have any other questions, ma'am. Thank you so much for this wonderful session. Now it is over to you, PL
Thank
madam.
you so much. Thank you, and very heartfelt congratulations to all of you because you have arranged this webinar in this pandemic situation. I really am feeling very good to be a part of it. It's just an honor. So, yeah, definitely we'll keep in touch. We'll be with you and
Yeah,
let
sure.
me know. Thank you
Thank
so
you,
much, everyone.
Shinjini ma'am. Thank you, Devashmita ma'am. Now, we are at the end of today's webinar series. Firstly, I would like to thank our Honorable Principal, Dr. Shima Banerjee, for giving us the opportunity to organize this international webinar series. Thank you, ma'am. I would also like to pay my sincere thanks to Dr. Shamalina Goswami, IQAC coordinator, for her kind cooperation in conducting this webinar. On behalf of the Microbiology Department of Hooghly Women's College, myself Dr. Piali Guho, would like to extend my heartiest thanks and gratitude to our honorable speakers, Dr. Abhishek Bonik and Dr. Debashmita Mukhopadhyay, for their valuable speech. We are indeed grateful to both of you for taking some time from your busy schedule to respond to our invitation. Thank you, sir. Thank you, ma'am. I'd also like to thank Professor Chirantan Hajra, HOD Physics of Hooghly Women's College, for taking the entire responsibility to coordinate this webinar in this online platform. We are also thankful to Professor Satyajit Bishwas Barsar for his active support. I'd also like to thank all the faculty members and non-teaching staffs of Hooghly Women's College for their support to conduct this webinar. Lastly, I would thank all the participants of this webinar to show their patience and make this a successful one. With this, we are at the end of today's session of International Webinar Series. Next, we will be meeting on 22nd August 2020 at 2 p.m. in the same platform. Till then, stay safe and healthy. Thank you.